Welcome to Rad Onc Smart Review RadBio Edition. Whether you're deep in board prep or, just want to really lock down your understanding of cellular responses, today we're diving into two absolutely foundational topics in radiation biology.
Yeah, really essential stuff.
We're talking about those critical gates cells pass through before they divide, specifically the G2M transition checkpoint, and then that elegant, but sometimes tricky, program cells used to eliminate themselves.
Oh.
Apoptosis.
That's right. And understanding how cells control their progression through the cell cycle and how they decide, live or die after DNA damage from things like radiation. It isn't just textbook stuff.
No, definitely not.
It directly impacts how cancers respond to treatment and really how we think about making them more sensitive. So our goal today is to walk through the key players, the concepts, make sure they stick, and link them directly to why they matter clinically for you.
Exactly. Think of this as our focused review session on these essential cell fate decisions. Let's jump right into that first big topic, the G2M transition checkpoint.
Okay.
You've heard us talk about the G1S checkpoint before making sure the cell's ready to copy its DNA. But G2M is arguably even more critical when we think about radiation effects.
Yeah, I'd agree with that.
It's the very last stop before mitosis. Once a cell gets past this gate, it's committed to dividing.
And that makes the G2 phase, the period right before this checkpoint, absolutely vital for DNA repair. The cell's just replicated its entire genome in S phase, right? So G2 is its chance to proofread, fix any damage left over. If there's still significant damage or if replication isn't totally finished, the cell has to be stopped here.
Because forcing a cell with damaged DNA into mitosis?
Yeah, it's basically forcing it into what's often called mitotic catastrophe. It's potentially lethal.
Okay, so that brings us to the key molecular driver, the thing that pushes the cell into mitosis. That's the cyclin B and cyclin-dependent kinase 1 complex, right?
Yeah.
CDK1.
Yep, cyclin B CDK1, often just called MPF M phase promoting factor.
Right, MPF.
When MPF is active, it drives the cell into M phase. So controlling its activity, that's the heart of the G2M checkpoint.
And controlling it involves this sort of delicate dance of phosphorylation, adding phosphate groups.
Exactly. A balance. CDK1 has specific spots, residues that can be phosphorylated to inhibit its activity. And the main kinase doing that inhibitory job is Wee1 kinase.
Wee1.
Yeah.
I always think of Wee1 as hitting the brakes on CDK1. Where exactly does it put those phosphates?
Good question. Wee1 primarily phosphorylates a tyrosine residue, tyrosine 15 on CDK1, sometimes 3914 as well.
Okay, Ty15, maybe 314.
And adding those phosphates basically locks that cyclin B-CDK1 complex in an inactive sort of off state.
So even if cyclin B is there, bound to CDK1, if Wee1 has done its job, the whole complex is just stuck. It can't push the cell forward.
Precisely. And that buys the cell that critical time it needs in G2 to finish repairs, like fixing radiation-induced double-strand breaks.
Okay, that makes sense. And this connects back to the DNA damage checkpoints we've talked about before, doesn't it? Like ATM, ATR, check 1, check 2. Absolutely connects. When DNA damage happens, those checkpoint kinases, ATM, ATR, and then check 1, check 2, they get activated. And they, in turn, either boost Wee1 activity or inhibit its counterpart, basically making sure those brakes stay firmly pressed on CDK1.
Keeping the cell arrested in G2. Okay, so if Wee1 is the brake, we obviously need an accelerator to actually get into mitosis when things are clear. That's where the CDC25 phosphatases come in.
You got it. The CDC25 family, and for G2M, it's mainly CDC25C. These are the enzymes that do the opposite. They remove those inhibitory phosphates that Wee1 put on.
And another kinase called MyT1 also adds phosphates, right? So CDC25 removes those two.
Yes, MyT1 also contributes inhibitory phosphates, and CDC25 takes care of removing them from both Tyr15 and Thr14 on CDK1.
So like releasing the brake pedal. Exactly. Removing those phosphates fully activates the cyclin B-CDK1 complex, and once it's active, boom, CDK1 goes on a phosphorylation spree. It targets all sorts of proteins to trigger chromosome condensation, nuclear envelope breakdown, spindle assembly, all the big events of mitosis.
So let me recap the flow. Cells in G2, Wee1 keeps CDK1 quiet, damage happens, radiation maybe, checkpoint pathways fire up, they crank up Wee1, keep CDC25 down, brakes on hard, cell stays in G2 arrest.
Perfect.
Only when the damage is fixed or replication's complete, then CDC25 gets active again, pulls off the phosphates, activates CDK1, and lets the cell proceed into M phase.
It's a really elegant gatekeeping system. And, here's a keyboard tip. Really understanding this balance, Wee1 as the brake, CDC25 as the accelerator, is fundamental. Radiation activates pathways that tilt this balance towards the brake, causing that G2 arrest.
Which leads right into the clinical relevance, because lots of cancer cells, they have messed up G1 checkpoints, often because of P53 mutations.
That's a very common scenario.
So that makes them rely much more heavily on this G2M checkpoint to survive after chemo or radiation.
Exactly. So if you hit those cells with radiation and you add a drug that inhibits, say, CHEK1 or Wee1,
you knock out their safety net.
Right. You might force those damaged cells into mitosis before they've had time to repair,
leading to that mitotic catastrophe we mentioned, lethal division.
Precisely. So targeting the G2M checkpoint is a major strategy being explored to sensitize tumors to radiation. Knowing these core players, cyclin, BCDK1, Wee1, CDC25, and how the damage response pathways hook into them, it's really essential stuff.
Okay, so that's how the cell controls when to divide. Very tightly regulated. But what if the damage is just too severe? Or maybe the cell is just old or not needed anymore. It doesn't just crumble messily, right? There's a programmed way.
Right. It doesn't usually just fall apart in that messy inflammatory way we call necrosis. Instead, cells have this built-in, genetically programmed self-destruct mechanism called apoptosis.
Programmed cell death. Sounds very orderly.
It is. It's highly regulated. The cell basically dismantles itself from the inside out very neatly. You see some characteristic changes.
Like what?
Well, the cell shrinks down. The DNA inside the nucleus condenses and breaks into fragments. The cell membrane starts bubbling. We call that blebbing. And eventually, the whole cell breaks apart into these neat little packages called apoptotic bodies.
Apoptotic bodies. And these get cleaned up.
Yeah, they're quickly gobbled up by phagocytic cells, like macrophages. Think of them as the cellular cleanup crew. And crucially, this happens without spilling the cell's guts and causing inflammation in the surrounding tissue. It's a very clean exit.
Okay, clean exit. And the key executioners, the molecules doing the work here, are called caspases.
Caspases, exactly. They're a family of protease enzymes that cut other proteins. Specifically, there are cysteine proteases that cleave their targets after specific aspartic acid residues.
Cysteine proteases cleave after aspartic acid. Got it.
And they're made as inactive precursors, called procaspases. They need to be cleaved themselves to become active. There are two main ways to kick off this caspase cascade and start apoptosis: the intrinsic pathway and the extrinsic pathway.
Intrinsic and extrinsic. Let's start with intrinsic. Sounds like it starts from inside the cell.
That's right. The intrinsic pathway is often triggered by signals from inside the cell. Things like severe DNA damage, very relevant for us, or loss of growth factors, maybe stress in the endoplasmic reticulum.
The radiation-induced DNA damage is a big trigger here.
A major trigger, yes. And a critical sensor for that DNA damage is the tumor suppressor protein P53.
P53 again, guardian of the genome.
Indeed. When P53 gets activated by DNA damage, one of its jobs can be to ramp up the production of several pro-apoptotic proteins. Some really important ones that boost belong to the Bcl-2 family specifically, BH3-only proteins like PUMA and NOXA.
PUMA and NOXA, induced by P53. And they're part of this Bcl-2 family, which seems absolutely central to this intrinsic pathway.
They are the gatekeepers, really. The Bcl-2 family proteins decide whether the mitochondria commit the cell to apoptosis. It helps to group them into three main functional types, based on which Bcl-2 homology or BH domains they have. These BH domains are just parts of the protein that let them stick to each other.
Okay, three groups based on BH domains. What's the first group?
First, you've got the anti-apoptotic or pro-survival members. These are the ones trying to prevent apoptosis. Think Bcl-2 itself, Bcl-STEC cell, MCL1, BCLW-A1. They generally have all four BH domains, BH1, 2, 3, and 4.
So Bcl-2, Bcl-XL, the good guys. From the cell survival perspective, what do they do?
Their main job is to bind to and inhibit the pro-apoptotic members of the family, keep them in check.
Okay, makes sense. Group 2.
Group 2 are the pro-apoptotic effector proteins. These are the ones that actually do the deed at the mitochondria. The main players are BAX and BAC. They have BH1, BH2, and BH3 domains.
BAX and BAC. The effectors, what do they do when activated?
When they get activated, they oligomerize, meaning they clump together, and they form pores or channels right in the outer membrane of the mitochondria.
Oh, they poke holes in the mitochondria. Okay. And the third group?
The third group are the pro-apoptotic BH3-only proteins. These are kind of the sensors or activators. Examples include BID, BAD, BIM, and those ones P53 induces, PUMA, and NOXA. There are others too, like HARC, BMF, BIC, As the name says, they only have the BH3 domain.
So just the BH3 domain. And they sense stress signals, like DNA damage triggering P1OX.
Exactly. They get activated by various death signals, and their job is upstream of Bax. They don't form pores themselves.
So what do the BH3-only proteins do then? How do they connect the signal to Bax?
They work in basically two ways. Some BH3-only proteins can directly bind to Bax and flip them into an active conformation.
Okay, direct activation.
Others work indirectly. They bind to the anti-apoptotic proteins like Bcl-2 or Bcl-XL. By binding them up, they essentially pull them off Bax, freeing Bax to become active. Think of it like distracting the guards.
Got it. So, let me see if I have this right. Healthy cell. Anti-apoptotic proteins like Bcl-2 are grabbing onto Bax, keeping them quiet.
Yep, holding them in check.
Death signal comes in, maybe DNA damage activates P53, which makes PUMA. PUMA is a BH3-only protein.
Right.
PUMA then either directly turns on Bax or it grabs onto Bcl-2, distracting it, which then lets Bax go free.
You nailed it. Either way, the result is active Bax.
Active Bax then lead to that crucial event you mentioned.
Yes, mitochondrial outer membrane permeabilization. MOMP.
MOMP. The point of no return.
Pretty much for the intrinsic pathway. Once Bax form those pores in the outer mitochondrial membrane, stuff starts leaking out.
And what's the most critical molecule that escapes from that inner membrane space into the main part of the cell, the cytosol?
That would be cytochrome C.
Cytochrome C, normally involved in respiration, right?
Yep.
But here it's a death signal.
Exactly. When it escapes into the cytosol, it plays a completely different role.
What does it do out there?
In the cytosol, cytochrome C finds and binds to a protein called APAF1. That's apoptotic protease activating factor 1.
APAF1?
Usually APAF1 is just floating around, kind of folded up. But when cytochrome C binds it, and usually with some energy from ATP or DATP, APAF1 changes shape.
Yes.
It unfolds a bit and starts clumping together with other APAF1 cytochrome C units.
They oligomerize.
Right. They form this large wheel-like protein complex called the apoptosome.
Apoptosome. Sounds important.
It is. It's the activation platform for the main initiator caspase of the intrinsic pathway.
Which one is that?
That's caspase 9. Procaspase 9 gets recruited to the apoptosome structure. Being brought close together on this platform allows the procaspase 9 molecules to basically cleave and activate each other through a process called autoactivation.
So the apoptosome brings procaspase 9 together, leading to active caspase 9.
Precisely. And now you have active caspase 9, the initiator caspase for the intrinsic mitochondrial pathway, ready to pass the signal on.
Okay, wow. So intrinsic pathway starts with internal stress, often DNA damage sensed by B53, leads to activation of BH3-only proteins like P1OX. They overcome the anti-apoptotic Bcl-2 family members, unleashing Bax, causing MOMP. Cytochrome C leaks out, binds FF1, forms the apoptosome, which activates caspase 9.
That's a perfect summary. You got the core steps.
Great. Now, what about the other pathway, the extrinsic one?
Right, the extrinsic pathway. This one, as the name suggests, is triggered by signals from outside the cell.
External signals. Like what?
Specifically, it's triggered by the binding of certain molecules called death ligands to death receptors on the cell surface. These receptors are part of a large family called the TNF receptor superfamily.
Death ligands binding death receptors. Okay. Can you give us some key examples we should definitely know?
Sure. The classic ones are the FAS receptor, which is also called CD95 or APO1. It binds to FAS ligand or FASL.
FAS and FASL. Got it.
Then there's TNF receptor 1 or TNFR1, which binds to the cytokine TNF alpha.
TNFR1 and TNF alpha.
Okay. And also the trail receptors. There are a couple, DR4 and DR5, which bind to a ligand called trail. That stands for TNF-related apoptosis-inducing ligand.
Okay. FAS, TNFR1, trail receptors. So one of these death ligands binds to its matching receptor on the cell surface. What happens next inside the cell?
Well, the ligand binding causes the receptor molecules to cluster together, usually in groups of 3. They trimerize.
Trimerization.
This trimerization brings together specialized regions on the inside part of the receptors called death domains. These clustered death domains then act as docking sites for intracellular adapter proteins that also have death domains.
Adapter proteins. Like FAD.
Exactly. FAD, FAS-associated death domain protein, is a key adapter, especially for the FAS receptor pathway. FADD binds to the clustered receptor death domains.
Okay, so receptor chromerizes, recruits FADD. What does FAD do?
FADD itself has another type of domain called a death effector domain, or DEED, and it uses this DDD to recruit the initiator caspases for this pathway.
Ah, so FAD brings in the caspases. Which ones?
It primarily recruits procaspase 8, sometimes procaspase 10 as well, which is very similar. They both have DEDs that allow them to bind to FADD's DED.
Okay, so now you have this whole complex forming at the membrane.
Yeah.
The trimerized receptor, the FADD adapter, and procaspase 8 all stuck together.
Right. And that whole assembly is called the DISC, the death-inducing signaling complex.
The DISC.
Wow.
Okay. And I bet being crammed together in the DISC is important for activating caspase 8.
You bet right. Just like with caspase 9 on the apoptosis, bringing multiple procaspase 8 molecules close together within the DISC facilitates their autocleavage and activation. They essentially activate each other.
Generating active caspase 8, the initiator for the extrinsic pathway.
Exactly, active caspase 8. Now, it's worth a quick mention here. There's an inhibitory protein called FLEPE.
FLEPE.
Yeah, FLICE, inhibitory protein. It looks a lot like procaspase 8. It has the DED, so it can get recruited into the DISC, but it lacks the actual protease activity. So it acts as a competitive inhibitor, blocking caspase 8 activation. Just another control point.
Fascinating. Always checks and balances. Okay, so now we have two initiator caspases potentially active. Caspase 9 from the intrinsic pathway triggered inside the cell
via the apoptosis.
and caspase 8 from the extrinsic pathway triggered by external ligands.
Maybe the DISC.
Where do these paths meet? Do they target the same things?
They converge. Both active caspase 9 and active caspase 8 have the job of activating the next level of caspases, the executioner caspase.
The executioners. These are the ones that actually dismantle the cell.
Yep, they carry out the demolition work.
And the main executioners we need to know are
caspase 3, caspase 6, and caspase 7.
3, 6, and 7, the executioner trio.
That's them. The initiator caspases 8 and 9 cleave the inactive proforms of caspases 3, 6, and 7, turning them into active protease. And once these executioners are active, they go to town, cleaving hundreds of different proteins throughout the cell.
And that cleavage leads to all those visible signs of apoptosis we talked about earlier, the shrinking, blebbing, DNA fragmentation.
Exactly. The executioners target key structural and functional proteins.
What are some really important substrates they chop up that are good to remember for exams?
Okay, key targets. Nuclear lamins. These proteins form the structural meshwork inside the nuclear envelope. Cleaving them leads to the breakdown of the nucleus and condensation of the chromatin.
Nuclear breakdown.
Another critical one, ICAD. That stands for Inhibitor of Caspase-Activated DNase.
ICAD. So it inhibits a DNase.
Right. Normally, ICAD keeps a DNase called CAD tightly bound and inactive. When executioner caspases cleave ICAD, CAD is released.
And the release CAD does what?
CAD then goes into the nucleus and starts chopping up the DNA between the nucleosomes, those protein spools the DNA is wrapped around. This creates DNA fragments of characteristic sizes, which produces that classic DNA laddering pattern you see on an electrophoresis gel.
Ah, the DNA ladder. That's a hallmark of apoptosis. High yield, you said.
Absolutely high yield. Remember, ICAD cleavage releases CAD, which causes DNA laddering.
Okay, what else do executioners cleave?
They cleave PRP, poly-ADP ribose polymerase. PRP is involved in DNA repair. Cleaving it impairs the cell's ability to fix damage, sort of ensuring the death process continues.
Takes out the repair crew.
Pretty much. And they also target cytoskeletal proteins like actin and gelsolin. Cleaving these disrupts the cell's internal scaffolding, leading to cell shrinkage, that membrane blubbing we talked about, and ultimately the formation of those neat apoptotic bodies.
So the executioners, caspases 3, 6, 7, systematically take the cell apart by cleaving lamins, ICAD, PRP, cytoskeleton. Very organized destruction.
It really is. And, these pathways aren't totally separate silos. There's actually an important link, a crosstalk between the extrinsic and intrinsic pathways.
Oh, how do they connect?
Well, active caspase 8, the initiator from the extrinsic pathway, can actually cleave one of the BH3 only proteins we discussed earlier, a protein called BID.
Caspase 8 cleaves BID.
Yes. This cleavage creates a shorter activated form called truncated BID, or TBID.
TBID.
And TBID then travels to the mitochondria and acts like a potent BH3-only protein. It directly activates Bax and Back.
Whoa. So the extrinsic pathway through caspase 8 and BID cleavage can directly fire up the intrinsic pathway at the level of Bax and Back.
Exactly. This essentially amplifies the death signal, especially in certain cell types. It ensures that even if the initial extrinsic signal isn't super strong, it can recruit the powerful mitochondrial pathway to make sure the cell dies.
That's a really important connection, the BID crosstalk. Good to remember. Wow. Okay, that's a lot of protein names, especially in that Bcl-2 family. You had that analogy earlier. Maybe we can revisit that to help lock it in.
Yeah, let's try that again. It can help visualize that balance at the mitochondria. Think of the mitochondrial outer membrane like a fortress wall, and MMP is like opening the main gate. Inside are things you want to keep in, like cytochrome C.
Okay, fortress wall gate, cytochrome C inside.
The anti-apoptotic proteins Bcl-2, Bcl-XL, they are the guards standing right at the gate. Their job is to physically hold on to backs and back the potential gate openers, preventing them from acting. They keep the fortress intact.
Guards keeping the gate openers restrained.
The pro-apoptotic effectors, backs and back, they are the gate openers themselves. If released or activated, they have the ability to assemble and form the actual channel or pour in the wall the open gate, letting cytochrome C escape.
Okay, backs and back open the gate if they're allowed to.
And finally, the pro-apoptotic BH3 only proteins, PUMA, NOXA, BID, BIM, etc. They are the attackers or the messengers carrying the open the gate order. They sense the danger signals. Their job is to neutralize the guards by binding the anti-apoptotic proteins or directly activate the gate openers, backs.
So the attackers take out the guards, or they directly tell the gate openers to act?
Exactly. If the attackers overwhelm the guards or directly activate the openers, then backs and back are free to form the pores, the gate opens, MOMP happens, and cytochrome C is released. Game over for the cell via the intrinsic pathway.
That analogy really helps clarify the roles. The guards, Bcl-2, trying to stop the gate openers, Bach, while the attackers, BH3 onlys, try to disable the guards or activate the openers. It's a constant struggle.
It is, and understanding that balance is absolutely key, especially when we think about cancer therapy. Now, stepping back, why does digging into all this molecular detail matter so much for radiation oncology residents?
Well, because radiation's primary effect is causing DNA damage, right? And as we've just discussed, that damage is a major trigger for both G2M arrest and the intrinsic apoptotic pathway via P53, Q-monoxA, and the whole Bcl-2 family drama.
Exactly. So a tumor cell's fate after radiation often comes down to how well it can handle that damage signal. Can it effectively arrest in G2M and repair? Or if the damage is too bad, can it successfully execute apoptosis?
And if it can't, if it's really good at arresting and repairing,
Right.
or if it has defects that make it resistant to apoptosis, maybe it overexpresses those guards like Bcl-2, or it has mutated P53 so it can't make the attackers PUMA and NOXA.
Then it's going to be much harder to kill with radiation. It basically ignores or overcomes the death signals. It becomes radioresistant.
And this understanding is precisely what's driving these newer therapeutic strategies we're hearing about.
Absolutely. We talked about G2M inhibitors trying to force cells into mitotic catastrophe. On the apoptosis side, if a tumor is resistant because it's blocking cell death, maybe we can give drugs that specifically counteract those blocks.
Like targeting the Bcl-2 family directly.
Exactly. That's where drugs called BH3 mimetics come in. They're designed to mimic the action of those pro-apoptotic BH3 only proteins.
So they act like the attackers in your analogy.
Precisely. A key example is venetoclax, pronounced VEH-nitoclax. It specifically binds to and inhibits the anti-apoptotic protein Bcl-2.
It takes out the guard.
Right. By inhibiting Bcl-2, venetoclax frees up pro-apoptotic proteins like BAX and BACIT, tipping the balance towards MOMP and apoptosis. These drugs are showing real promise, particularly in blood cancers, where Bcl-2 is often sky high, and they're definitely being looked at in combination with radiation for solid tumors, too.
That's fascinating. Are there other ways people are trying to target apoptosis?
Oh, yeah. There are strategies looking at inhibiting proteins that block the caspases themselves, things called IAPs, inhibitors of apoptosis proteins. XIAP is a major one, so IAP inhibitors are being developed.
Taking the brakes off the executioners.
Kind of, yeah. Or people are trying to directly trigger the extrinsic pathway using things like synthetic trail ligands or antibodies that activate the trail death receptors specifically on cancer cells.
So lots of different angles try to push resistant cells towards apoptosis.
Exactly. And understanding the status of these pathways in a specific patient's tumor, does it have high Bcl-2, is P53 mutated, could become really important for predicting who might benefit from these targeted approaches alongside radiation. These could be a predictive biomarker.
This whole area is built right on top of the fundamental mechanisms we just walked through.
Okay.
Fantastic. Let's try and boil this down into some really concise clinical pearls for board prep.
All right, clinical pearls. Pearl number 1. Remember, the G2M checkpoint is a critical defense after radiation damage. Inhibiting key players like Wee1 or Chek1 is a strategy, especially in p53 mutant cancers, to force cells into lethal mitotic catastrophe and boost radiation effectiveness.
Okay, G2M inhibitors for radiosensitization via mitotic catastrophe. Pearl 2.
Pearl number 2. A tumor's inherent ability to undergo apoptosis is a major factor in its radiosensitivity. If a tumor resists apoptosis, maybe high Bcl-2, maybe bad p53, it's generally going to be more radioresistant.
Makes sense. Apoptosis potential equals radiosensitivity. Pearl 3.
Pearl number 3. Because of that, we're developing therapies that target the apoptotic machinery itself. BH3 mimetics, like venetoclax, blocking Bcl-2, aim to restore apoptosis potential and could enhance radiation response, especially in resistant tumors.
Excellent. G2M inhibitors apoptosis predicts sensitivity, and targeting apoptosis, like with BH3 mimetics, can overcome resistance. Great pearls. Now, speaking of boards, ready for a quick board blitz?
Let's do it. Fire away.
Okay, first question. Entry into mitosis is primarily driven by the activation of which cyclin CDK complex? A. Cyclin D, CDK4. B. Cyclin E, CDK2C. Cyclin A, CDK2. D. Cyclin B, CDK1.
Okay, that one's fundamental. The correct answer is D, cyclin B, CDK1. That complex, also known as MPF, is the master regulator driving the cell from G2 into M phase. You absolutely have to know that one.
Perfect. Question 2. Wee1 kinase inhibits the activity of the cyclin B CDK1 complex by A. Promoting the degradation of cyclin BB, phosphorylating and activating CDC25 phosphatase. C. Phosphorylating inhibitory sites on CDK1. D. Sequestering cyclin B in the cytoplasm.
Right, Wee1 is the brake. How does it work? The correct answer is C. Wee1 adds those inhibitory phosphates onto CDK1 itself, specifically tier 15 and thir14, keeping the complex switched off.
Got it. Question 3. The intrinsic pathway of apoptosis is initiated by the release of which molecule from the mitochondria into the cytosol? A. SMAC-Diablo, B. Cytochrome C, C. AFF apoptosis-inducing factor, D. Caspase 8.
Okay, intrinsic pathway trigger from mitochondria. While SMAC and AIF are released, the key initiator we focused on for apoptosome assembly is cytochrome C. So the correct answer is B. Cytochrome C release is that pivotal step leading to caspase 9 activation.
Exactly. Final blitz question. Which of the following proteins is a BH3-only protein that can be transcriptionally upregulated by p53 to promote apoptosis? A. Bcl-2, B. Bax, C. PUMA, D. Caspase 9.
All right, looking for a BH3 only activated by p53. Bcl-2 is anti-apoptotic. Bax is a pro-apoptotic effector. Caspase 9 is an initiator of caspase. PUMA fits the bill perfectly. It's a BH3 only protein, and its gene is a known transcriptional target of p53. So the correct answer is C, PUMA. NOXA is the other big one often mentioned.
Fantastic. Nailed the blitz. You know, while we're making really exciting progress targeting these pathways, it's not always straightforward in the clinic, is it?
Oh, absolutely not. There are definitely challenges. Tumor heterogeneity is a huge one. You might have subclones within the same tumor that have different mutations or expression levels, so a targeted drug might work on some cells but not others.
Right. Resistance can develop.
Exactly. Cancer cells are incredibly adaptable. They can find ways around these therapies, maybe by upregulating a different anti-apoptotic protein if you block Bcl-2, for example, and figuring out the best combinations. Should you give a checkpoint inhibitor with a BH3 mimetic? How do you sequence them with radiation? Those are really active areas of research right now.
Yeah, the complexity, especially within that Bcl-2 family with all the interactions, it's daunting.
It is. Simply blocking one protein might not be enough if the cell can compensate elsewhere. It really highlights why deeply understanding these pathways is so crucial for developing smarter therapy.
Definitely. Okay, so let's just quickly summarize the absolute key takeaways from today.
Sounds good. First, the G2M transition. It's the gate before mitosis, tightly controlled by cyclin BCDK1 activity. Remember, Wee1 puts on the inhibitory phosphate brakes, CDC25 phosphatase takes them off to accelerate entry into mitosis, and DNA damage checkpoints reinforce the braking.
Got it. Then apoptosis, programmed cell death, two main paths. The intrinsic pathway, triggered by internal stress like DNA damage, hinges on that Bcl-2 family tug-of-war at the mitochondria, leading to MOMP, cytochrome C release, apoptosome formation with APAF1, and activation of initiator caspase 9.
Perfect. And the extrinsic pathway, triggered by external death ligands binding death receptors. This leads to DISC formation involving receptors, adapters like FADD, and initiator caspase 8 activation at the membrane.
And both pathways ultimately converge on activating the executioner caspases 3, 6, and 7, which are the ones that actually dismantle the cell by cleaving key substrates. And don't forget that crosstalk where caspase 8 can cleave BID to amplify the signal through the intrinsic pathway.
Exactly. And the big picture relevance for you, these checkpoint and death pathways are fundamental determinants of how cancer cells respond to radiation. Understanding them is key to understanding radiosensitivity and developing strategies, like G2M inhibitors or BH3 mimetics, to overcome resistance.
Mastering these molecular details and seeing how they connect to clinical problems and therapies is just so vital as you prepare for your boards and, for your whole career in radiation oncology.
It really is. It might seem like a lot of detail, but building this strong foundation now will serve you incredibly well. You can definitely do this.
Absolutely. Keep reviewing, keep connecting the dots to continue practicing and really solidifying these concepts. You can find practice oral boards over at radoncSmartLearn.com.
And be sure to subscribe to Rad Onc Smart Review for our next deep dive into radiation biology.
Thanks so much for tuning in.